Welcome to meiosis. Meiosis is the purpose of producing gametes, which are also called sperm and eggs. The start of meiosis is in interphase 1, which starts off with a diploid cell. In G1, the cell starts to grow. In S phase, the DNA is copied and replicated to make identical genetic information, and G2 is when the cell is prepared to start dividing. Prophase is where the chromosomes are becoming visible, and the cellular membrane around the nucleus is starting to break down to allow the chromosomes out into the cell. This is what a chromosome looks like. You have a diploid chromosome with a centromere in the middle connecting the two sister chromatids on the outside. Once they become diploid chromosomes, they line up with their homologous pairs and will start to cross over each other, also known as synapsis. Synapsis is the pairing of two homologous chromosomes, which allows them to match up prior to segregation, which makes them possible for chromosomal crossover. When crossing over, there is a point of contact called chiasma. At this point of contact is where genetic material is exchanged, allowing the chromatids to cut off pieces of themselves and swap them so the genetic information is unique, also known as genetic recombination. Now we're in metaphase. In metaphase, the homologous pairs start to line up against the equator, which allow each pole of the cell to release spinal fibers that then attach themselves onto the centromeres of the chromosomes. Independent assortment ensures the random information of chromosomes. This makes sure uniqueness and variation among species is stable. Next is anaphase. The spinal fibers start to pull the diploid chromosomes to other ends of the cell. One bad thing that can happen in anaphase is non-disjunction. This is when the homologous pairs don't separate properly, as instead of splitting apart, they go together in one direction, which leads to bad variation. This can occur in anaphase 2 as well. The normal way is called disjunction, and that is when everything separates properly and all gametes are left with an even amount of chromosomes. Telophase 1. Telophase 1 is the separating of the outer cell membrane in order to form two daughter cells, which are preparing it to undergo the second stage of meiosis, which is dividing remaining chromosomes. Meiosis actually used to be called reduction division, because the first stage of meiosis focuses on the reduction of the amount of chromosomes in the cell to prepare it for the second stage, which is dividing, as it is dividing the remaining chromosomes in 4 haploid gametes. To start, let's go back to prophase, which is a diploid chromosome that starts showing up after condensing from chromatin. Next is metaphase, which is when these diploid chromosomes line up against the equator once again and the spindle fibers are recreated to then attach themselves back into the centromeres to separate them. In anaphase, the spinal fibers pull the diploid chromosomes to each end of poles, creating haploid chromosomes, which is a single strand or a single chromatid that is not attached to anything. Telophase 2 is a complete and final stage before cytokinesis. This is when the cytoplasm is separated and the nucleus reforms itself to protect the chromosomes. Cytokinesis is when 4 haploid daughter cells is created, which are germ cells. When egg cells does not divide evenly during telophase 2, there is 3 polar bodies shown. A polar body is a small haploid cell which does not possess the ability to undergo fertilization. However, the cell with more cytoplasm becomes a mature ovum. Thanks for listening, and I hope you started to learn about meiosis.